Okay, let's get started for the second discussion on muscle. I also want to draw your attention to the syllabus. So if you would please take out your syllabus. What color were we this semester? Salmon? Orangey salmon color. And we are going to, when we get back from break, we're going to have a lecture on 3:10. That's a Tuesday, right? Thursday, 3:12. And then we have spring break before that. And then we're going to have lecture on Tuesday, 3:17 and 3:19. I've been looking ahead, and based upon where we are, I think I'm going to have to do the domino thing. I really hate doing this, but I think the blood conversation makes a whole lot more sense being in this next test than not being in this next test. So here's what we're going to do. Today, whatever today is, we're going to finish up muscle. Then, very soon, very, very soon, I am going to record 2 lectures on heart. And they will be for these 2 dates. These will be cardiovascular system. Okay? And then I just found out the other day that on those 2 dates, I've been invited to go down to Miami for a planning session. And so I will likely, unless something changes, would not be here for those 2 lectures. They will be on YouTube, and I'm going to try to get those done sooner than later so that you don't have to wait until the 10th and the 12th to listen to that material. If over spring break you wanted to, you could. If you don't want to, you don't have to. But I will post those earlier. Then, this would be the day that I would do the blood lecture. The problem with that, I think, is that it's getting too close to the day of the test, right? That's worrisome, right?
That's the day of the test.
Well, I'm moving it to the 19th. I didn't say that yet, right? So I'm going to move it. I want blood to be on this test. So I'm going to move the test to the 19th. But I always hate it when I'm presented with material really close to the test, right? Because blood, we've had some information on blood, but we don't know a lot about it. So what I'm going to do is I'm going to create all 3 of these lectures, okay, over the next few days. And you're going to have a cardiovascular part 1, a cardiovascular part 2, and a blood lecture. They will all be on YouTube. I'll send the links. I'll send instructions. Then on the 17th, right, because I won't be here the 10th or the 12th, we won't have lecture. You'll still have labs. We got the labs covered, so labs won't change. But we won't have lecture. On the 17th, we'll have a review session. So on the 17th, right, come on in, and by then have listened to the cardiovascular talk and the blood talk, and then on the 17th, rather than having a lecture, we'll have a review session. And I think that's a good way to approach this. I don't want you to feel out of place, not able to ask me questions about things, and then on the 19th, we'll have the test. Okay? Does that seem reasonable? I don't like being away. It doesn't happen very often. It's probably the first time in 6 years where I've missed a couple of lectures in a row. And but I am very thankful that we have some technology to help us out, so that I would rather you listen to YouTube than have another instructor come in here and give you information, and it just isn't as consistent, is the way I approach things. So that's the way I have thought through this. Are there any concerns about that? So the next test would be over muscle, cardiovascular, and blood. It would be on the 19th of March. I won't see you on the 10th or the 12th. On the 17th, we'll have a review session. So you'll have 3 lectures that you will listen to on your own between now and the 17th. That should be enough time to get 3 lectures out of the way. What do you think? Okay. I've also already put the homework assignments up. They're on mastering. And I will provide at least 2, maybe 3 quizzes. So I've already put up a muscle quiz, and I will put up a cardiovascular quiz, and I may or may not put up a blood quiz. There will be 12 to 14 of those quizzes before we're done in the semester. Only 10 will count. So if you've missed one, didn't do well on one, missed a couple, you know that you'll be able to drop those lowest grades at the end of the semester. So if you missed a quiz, do not fret. There will be some extra ones to make up that 0 or that low grade. Does that make sense for everyone? Any concerns for me? Just change the test date from the 17th to the 19th and be watching for an email. if you're going away on spring break, enjoy yourself. Just go. I mean, we all need the rest. We need the revitalization. But if you are able to, between now and the 17th of March, make sure you've listened to the 3 lectures and do look at mastering for the assignments. I've got homework assignments and I've got the quizzes. And please just look at those dates. I won't change them once they're there. I won't change them on you. But don't miss those dates. And then on the 17th, we'll have a review session for the entire hour and a half. We'll just be here to answer questions and talk about things. And then on the 19th, we'll have the exam. Fair enough? Was that question?
Case studies.
Case studies, I did put out a link on those. They will be due on the, what did I say? 12th. Yep. So they'll still be due on the 12th. Okay. So you can bring those to my office, bring those to my secretary, bring those to Mr. Mueller that evening on Thursday night. If you haven't done earlier, that's fine, right? But you can send all that stuff in, and I will send out another clarifying email, but each group will also upload their questions to SafeAssign, and just one person from each group will do that. So you'll get some instructions on that from me. You'll also should be getting some instructions from Mr. Mattoni on uploading your lab paper to SafeAssign before we see each other again a couple weeks from now for lab. So I'll be watching for that. So whoever is collating your paper, putting together the final version, and is going to print it to hand it in, will be the same person who will just simply upload that to SafeAssign. It'll take about 30 seconds to do that, and that'll go into the anti-plagiarism tool that we have here at the college. Okay, so please make that a change to your schedule, and hopefully that is the last of changes. But I'm excited about this opportunity, and I appreciate your support in that. Let's review. Last time we talked about skeletal muscle, and we talked about thick and thin filaments, actin and myosin. We said that the thin filament is not just actin, but it's actually a helical molecule of actin along with some tropomyosin going along the length of it, some little troponin molecules. On the tropomyosin, there was also a protein called actinin that connected it all together, and nebulin also bound the actins, the G-actins together. Then we also looked at myosin, and we said that myosin was not only the myosin molecule, but also that big molecule called titin that sort of went through the middle of it like a big spring for like a big trampoline. So let's try to put this whole story together. And this image is in your textbook. It's 9.8. I know it's small there for you. But let's kind of talk about this big idea, and by doing so, it'll be a good review. So on this image, action potential is sent. We're going to muscle, so you tell me, where is that action potential initiating? That signal to cause your skeletal muscle to move was initiated in your frontal lobe at your motor strip, right? And that signal is going to travel down your motor neuron, down the spinal cord, down that tract, from your spinal cord out to your muscle. As it gets down to the end of that axon, at the axon terminus, or at the synaptic knob, what's going to happen? Somebody start talking to me. That voltage signal is coming down the axon. It will trigger what? This is old. Calcium. Yeah. Voltage-gated calcium channels to open, and calcium will come rushing in and will bind to synaptic vesicles, which will be filled with a neurotransmitter called acetylcholine. That acetylcholine will now release itself into the synaptic knob, or into the synaptic cleft. Now, when we were learning about this earlier, the next cell in series would have been another neuron. But now we're talking about muscle. So now we're talking about the muscle being the postsynaptic cell. The portion of the axon is going to touch the muscle. What do we call that part of the muscle? There's a name for that part of the muscle where the axon terminus comes in, called the motor end plate. Imagine the electrode is like, imagine the axon is like a little electrode, and it comes down and it kind of sits on a part of the sarcolemma, the muscle cell, and that area where it sits is called the motor end plate. What is found in the motor end plate? On the sarcolemma, what are we going to find there? Receptors for acetylcholine, right? And the acetylcholine is going to bind to those receptors. Those are the same receptors we talked about before. They are ligand-gated channels. And when the acetylcholine binds, it's going to open up the channel. That's going to allow sodium to do what? What does sodium always do? Goes into a cell, right? It's going to depolarize the muscle cell. And at this point, an action potential will be generated. And that action potential will run along the sarcolemma and will dive down into those rabbit holes called T-tubules. We know that the T-tubule is in close proximity with two sacs, part of the sarcoplasmic reticulum called the terminal cisternae. And we have this triad. We've got the T-tubule down the center and the two sacs of sarcoplasmic reticulum. That signal coming down the T-tubule is going to activate what? Voltage-gated calcium channels, similar to the ones we had back in the axon terminus. And they're going to open up, and calcium is going to go flooding out from the sarcoplasmic reticulum into the sarcoplasm, into the cytoplasm of the muscle cell. What is it that's stuffed inside a muscle cell? What special organelle? Myofibrils. Remember, the myofibrils run the entire length of the muscle cell, and there's hundreds of thousands of them bound in there. And we know that the myofibrils are themselves made up of little units called sarcomeres. And the sarcomeres are a collection of thick and thin filaments overlapping each other. What is that calcium going to do? That calcium that was released by the sarcoplasmic reticulum is going to bind to a protein called troponin. Troponin is that little snowman molecule attached to the tropomyosin on the thin filament. By calcium binding to troponin, it's going to cause that entire tropomyosin molecule to shift. The shifting of that is now going to allow myosin to bind to actin. The tropomyosin was blocking that previous to that. So now the actins and myosins can bind. The actin and myosin binds, and the myosin head does a little shift. They're still attached. The actin and myosin are still attached. What is necessary for the actin and myosin to dissociate, to separate? ATP. So it takes ATP to separate the actin and the myosin. Once we separate, right, because what did that myosin head? That myosin head includes an enzyme activity called ATPase. So the myosin head can cleave that ATP and use that energy to recock. And then it'll bind actin, and then power stroke, and then ATP again to separate. This is happening thousands of times along every myosin-actin interaction. As long as there is an action potential being sent, an acetylcholine going across the cleft, and as long as there's calcium being released by the sarcoplasmic reticulum bound to the troponin, the tropomyosin will stay out of the way. The actins and myosins will do their little sliding filament dance, and as long as there's ATP around, it'll just keep on happening. If there's any problem along the way, it could be a problem with acetylcholine, it could be a problem with calcium, it could be a problem with ATP, then that could affect and interfere with nerve to muscle transmission. Okay? Is that a nice little quick review? I was speaking with some students today, and they understood pretty well the little story about calcium and troponin, and they understood action potentials, but they hadn't yet been able to bring it all together into one story. Okay? You need to be able to do that. We cannot forget about the action potentials that we've already talked about in the past. You'll be reminded of action potentials this week in lab through a little activity on PhysioX. So I think this is all going to come together at the right time for you, but please don't, try not to let this story get too stale in your head over break. If you can hold on to that story, how action potentials, and what do we call that? Excitation coupling, right? If we can get the excitation of the muscle cell to couple over to the contraction with the calcium, then I think you'll be in good shape moving forward. Questions on any of that process? That was through 9.7. Anything to clarify? Yes, sir.
What kind of receptors on the muscle
It's acetylcholine. It's a ligand-gated sodium channel.
Okay, so is it like a nicotinic receptor or muscarinic?
It is going to be, let's not worry about that yet. Okay? It's acetylcholine, so it's going to be a cholinergic. receptor, right? But let's not worry about the other right now. We haven't discussed that in a while. Okay. Any other things to clarify? If you can't tell that story to your friend, practice it. Okay, just going from action potential all the way through to muscle contraction, and make sure you can name the players along the way. So, having said this story, what's the connection between excitation, contraction of the muscle and muscle tension? The muscle is going to shorten, isn't it? That's going to increase the tension on the muscle. So the thick and thin filaments are going to slide over each other, and the sarcomeres will all be pulled shorter. The Z discs will pull in shorter. Let's remind ourselves of that. A sarcomere is defined as going from Z to Z. Z is a protein, right? It's kind of a little squiggly line in all the cartoons. From Z to Z, end of the alphabet, the way I think about it, from end to end. And then the myosin, however wide the myosin is, that is called the what band? A band. Wherever the I bands are where the thin filament are, the actins. And then there's an M line right down the middle. Okay, so if you keep track of those, you'll be in good shape. So as the muscle is shortening, we're producing tension on the muscle, and we know that the muscle's attached usually to tendons on each end, and that's going to cause the skeleton to be pulled and moved in a predictable way. So again, be able to go through this story and think about, here's that same picture again, and go through the steps of how it is that we create the shortening. Now keep in mind, this is a little bit of a muscle cell, and each of those overlapping dark and light bands represent what? Pretty much another sarcomere, isn't it? So we know we have thousands of sarcomeres end to end, all shortening simultaneously in order to create the tension on the muscle, pulling on those tendons. So as the sarcomere is shortening, we're going to be increasing the tension. And as I said before, the amount of tension does not, I don't think I've said this, the amount of tension does not depend on the number of sarcomeres that are contracting. Because I want you to think about this. In a muscle cell, what do we know about muscle cells? They're extremely long, aren't they? If a muscle cell is activated anywhere along that entire muscle cell, then the signal will be going down the T-tubules, and calcium will be released, and troponin will be bound, and sarcomeres will shorten. So every sarcomere down the entire length is going to be stimulated and contract together. Either the muscle fiber is on or it's off, the entire fiber. We don't get to choose just a few sarcomeres on and a few sarcomeres off, right? So every sarcomere along the entire length of a muscle cell will either be on or off. That is, it's either contracting or it is relaxing. Now, there is an optimal resting length of a sarcomere. Okay, and I'll show you a picture of this in a moment. The actins and myosins at rest need to be overlapping enough to grab each other and slide. If they are too separated, they can't grab. If they're too far apart, we'll see what happens here in a moment. To get the most tension, there has to be an optimal distance or overlap of the actins and myosins. Here's, if the sarcomere is in its normal place, then the sarcomere can change from 75 to 130% of its optimal length. We have in our skeletal system, in our muscular system, ways to prevent too much stretching. We talked a little bit about those last semester. I'll mention them again along the way. Remember we had Golgi tendon organs, and we have ways of making sure the muscle's not being stretched too much. So we've got some reflexive control in place. I'll show you that picture in a moment. If the sarcomere is not optimal, then there can be a reduction in the tension, and this can be about tone. When you hear about someone is well-toned, what does that mean when you hear that? Someone is well-toned. their body's well-toned, their muscles are well-toned, what comes to mind?
Being in good shape.
Being in good shape, muscles having good form or definition. The greater the tone, that means that these little sarcomeres are constantly changing, right? There's an optimal distance, and the greater the tone of your muscles, that suggests that your muscles are constantly contracting a little bit, right? Someone who's well-toned has a little bit of contraction going on sort of in the background, if you will, keeping their muscles well-defined. That's also going to create a lot of energy being used, right? A person who's well-toned, a person who has more muscle is going to burn more ATP watching TV than I will, right? Because those toned muscles are constantly using energy as they are getting to that optimal length and helping define the muscle. Right? So people with more muscle have a higher basal metabolic rate. They burn more ATP. They utilize more ATP at rest than a person who has less muscle. Now, if the sarcomere is at an extreme length and there's no overlap between the actins and myosins, then no tension can be generated at all. And so you'll see what I mean by this when you see the graph in a moment. If the tension can also be, you can also have reduced tension due to having very little room. In other words, there's too much overlap. If there's not very much room for the actins and myosins to slide over, then you won't have much shortening of the muscle. So let's look at the graph, and then all these words will make sense to you when you look back at it. Here is a sarcomere at ideal resting position. Now what that means is that the actins and the myosins are overlapping sufficiently so at rest, so that when the muscle begins to contract, there's enough, if you will, grip between the actins and myosins that they can continue to slide over each other and shorten the sarcomere. What if, let's look at the two different examples. What if instead, looking at this picture, what if there wasn't much overlap between the actin and the myosins? If your sarcomeres were a little bit too far apart, look at my fingers, right? And rather than, let's say this is resting here, my sarcomere is here. They're overlapping, right? But they don't have very much grip, do they? So if I was to ask this sarcomere to grip, there wouldn't be much to start with. That's going to reduce the ability of them to slide over each other. What if they were completely separated? Right? If the actins and myosins, look at this picture, if the actins and myosins were completely not overlapping and calcium comes into the picture, what can those actins and myosins do? Nothing, right? They can't grab each other. They can't bind. So there's an optimal distance. If it's too far apart, it's not good. But what if over here it's too close? If the actins and myosins are starting already half split over each other, then when you go to contract, there isn't much more to go, is there? So then you wouldn't get as much force either. So there's a graph that represents this pretty straightforward, I think. Sarcomere, this is ideal right here. Okay, ideal, the maximum. Here's the normal range. We're talking around 2 micrometers. Somewhere in the 2 micrometer width is ideal for the entire Z to Z sarcomere. If the length is increased or if the length is decreased, what happens to the tension that can be generated? It drops off. Okay. So there's an optimum distance. That gives us the top of the curve. If the sarcomere is too short or too long, then the tension that can be produced is sacrificed. Does that make sense? Does that make sense? Everyone? Yes? Caprice, that makes sense? Okay. So that means all of your sarcomeres are at rest at an ideal distance. Now, when a muscle fiber contracts, we're going to call that a twitch. Okay? We got a few new words here. Muscle twitch is a single contraction relaxation sequence of a muscle fiber. Now, depending upon if this is cardiac muscle or smooth muscle or skeletal muscle, it can vary in its timing. It can vary in its location. It can vary in different conditions. But a muscle twitch will be a single little twitch of a single muscle cell. That's a muscle twitch. Okay. Now, the more tension that is created, we can put this on a graph, and we've seen these graphs before. A myogram, right, would show the tension of the muscle. We did an EMG, didn't we, in lab with the muscle physiology labs. We were looking at electromyograms and myographs and looking at this tension, not of a single fiber, but of the entire muscle, right, when we were looking at the EMGs in lab. Fasciculation is simply a term. Have you ever had an involuntary muscle twitch? Right? Maybe around your eye, little twitches sometimes, or you'll just notice something maybe on your forehead, just someplace, right? Little twitches are happening. That's called a fasciculation. It's just usually a contraction of a group of fibers controlled by one motor unit. Remind me, what's a motor unit? One axon and all the muscle fibers that it innervates, right? So one axon travels not to just one muscle cell, but to many, sometimes, and that's a motor unit. So it can involve more than one fiber, right, more than one cell, and it involves an entire motor unit. So let's talk about, we all had that little eye muscle thing happen. That is very, very quick, isn't it? Look at the timing here. It increases. So the eye muscle is very, very quick. Those are very fine muscles, small motor units, very fine control of the eye muscles. The gastrocnemius, sort of we'll call it a medium muscle, I suppose, takes a little bit longer to tense and relax. And if we're talking about like the soleus is a deeper muscle along your calf. That takes a lot longer to contract and to relax. Notice that the maximum tension is the same. Here we're just talking about time. So it takes a little bit longer for it to respond, and it takes longer for it to relax, whereas the eye muscle is very, very quick to respond. But regardless, those would all be twitches. It's one cell or one group of cells from a different part of your body responding. We can put the muscle twitch or the tension here into some phases. There's a latent phase, a latent period. What's a latent period?
Late period.
A what?
Slow.
Slow period, maybe kind of a period for things to get jump-started, to get going. So once the initial signal comes down from the neuron, there's going to be a little bit of a time lag, right? Because what has to happen? We know what has to happen, right? We got the acetylcholine, we got going across the cleft, we got the sodium channels opening, we got the calcium channels opening, we got the binding of the troponin. That all takes 2 milliseconds, right? It's pretty wild. But there's a little bit of a break, a little bit of a time lapse from the time the signal is received by the muscle cell and when it will actually begin to contract. During this time, there is no tension. Again, it takes time for the calcium to be released from the sarcoplasmic reticulum. Then the muscle will go into contraction phase. This is when the tension is going to begin. All the sarcomeres will begin to shorten. The calcium is binding to the troponin. The cross-bridging is occurring. And then finally, after the signal has dissipated, there's a relaxation phase. It's going to take a little bit of time for that calcium to be removed. It's going to take a little bit of time for the acetylcholine to be removed from the cleft. So there's a little bit of time at the end of the signal to relax. This is a lot longer than latency. The latency is about 2 milliseconds. The relaxation phase is going to be more like 25 milliseconds from the peak to complete relaxation. Okay. Again, you just got to wait for the calcium levels to drop off, for the tropomyosin to shift back in place, and for the actins and myosins to detach, and for the sarcomeres to return back to their original place. So we can look at this in a graph. Again, here's the tension. Here's time. This is all in milliseconds, faster than most of us can even imagine. In real life, we would say this is happening, almost simultaneously, right? So here's that little tiny latency phase. Just a couple of milliseconds here. So the signal has come in. There's the stimulus. A little bit of lag, and then the contraction phase. And from the maximum contraction to the end where we're back to normal, that relaxation phase, somewhere around 25 or so milliseconds. So latency, contraction, and relaxation. So just know those different names for the three phases of a contraction cycle. Now, how are we going to get the maximum? How are we going to get the maximum tension from a muscle? A muscle has thousands of cells. It has many axons involved, right? Your biceps brachii is not innervated by a single axon. You have many axons that send signals down to your biceps brachii. If you want to lift up something really heavy, you are going to, quote, recruit more motor units to fire. In fact, if you know the thing you're about to lift is the heaviest thing you've ever lifted, then you're going to voluntarily recruit every motor unit, and you're going to ask every fiber in your biceps brachii to attempt to shorten, right, for the maximum. If, however, you're just going to reach over and pick up a pencil, and you know from your experience that pencils weigh just a couple of ounces, it doesn't take a whole lot of muscle effort to do that, you're only going to, quote, recruit a limited number of motor units. Not every cell in not every fiber in your muscle is going to be working as you contract and lift up that pencil. So that's what we're getting at right now, this idea of recruitment, and how is it that we tell our muscles to, pick up something light versus pick up something heavy. So it's all going to come down to the amount of tension on each fiber, as well as the total number of fibers that you stimulate within the muscle. So there's 4 levels of tension. Okay, we're talking about here a single muscle fiber, and we're going to be looking at these 4 different levels of tension. Treppy, kind of a stair-step look. We'll see a wave, what's called wave summation. We'll then move into an incomplete tetanus and then complete tetanus. When you hear the word tetanus, what comes to mind? Lockjaw, right? And what is lockjaw? The muscle is completely tensed. It won't relax, right? It's doing maximum work, and unfortunately in tetanus it won't let go, right? If we were to have tetanus of our heart, we would die, right? Because our heart would stop. So we'll talk more about that when we get to cardiovascular system. But as long as you have a meaning for that word tetanus in your head, the muscle is tensed to its max, then this conversation will make good sense. So Treppy, it means staircase from the German, and basically you're just simply going to keep stimulating a muscle cell. It's going to contract, and then it's going to relax. Then it's going to contract, and then it's going to relax. Now what happens if you start doing it really fast? It will contract, but it won't be able to completely relax. Okay? So the faster you are sending an action potential down, telling a muscle to move, at some point, it never gets a chance to relax, and instead we'll see this Treppy picture, and I'll show this to you in a moment. And you get this stair step. Now, you can do this for about 30 to 50 stimulations. Most skeletal muscles do not demonstrate Treppy, only because we really can't, we're not going to be doing something as fast as this requires, but I'll show this to you in time. So here's a little graph, and over time, look what happens. See how this goes up? See how that's going up? So you contract the muscle, it goes up a little bit, and even before it has a chance to completely relax, you do it again, you stimulate it again, and again, and again, and again. Now what's going on inside the cell? For relaxation to occur, what must happen? Remember, there was a 25 millisecond relaxation phase, wasn't there? And during that 25 milliseconds, what has to happen? Calcium has to come off from the troponin. The tropomyosin has to shift back. The actins and myosins have to let go of each other. The acetylcholine, right, has to go back at the cleft. All that has to happen for relaxation to completely occur. If you keep sending a signal really fast, then it never gets to completely relax. Therefore, calcium never gets completely cleaned out of the act. So you never completely relax. And if you keep pushing the button or keep sending that signal, then you start seeing this increase, right? This stair-step effect over time because the muscle never can completely relax. Now, wave summation, same idea, right? You're just going to keep adding one twitch to another. And you're going to finally get a maximum value. And we'll see this little wave that you just saw, this wave summation. So now you really start seeing it going up. So here's a wave, 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 and you can see, change colors. You can see that we're just increasing and we're going up. Okay? Wave summation, you're adding up all the waves. At some point, you get to an incomplete tetanus. This is a rapid cycle. You're almost at peak. I mean, you're almost at peak. There's really no period of relaxation at all. There's a little bit, though. It's not completely at tetanus. So we'll call that an incomplete tetanus. So basically, you've gone through your little wave summation, and now you're up here at the very, very top. And you're not completely at tension, but man, you're way up here. Okay, there's still a little bit of relaxation going on. And then finally, you get to tetanus. And tetanus is when the muscle has no relaxation periods at all, at all, at all. So at complete tetanus, you are stimulating the muscle so rapidly, there's absolutely no relaxation phase at all. No calcium is even taken back up into the sarcoplasmic reticulum. It has no opportunity to do that. So you have peak tension for continuous contraction. And if this were to happen normally to your muscles, right, you could damage them. And this rarely happens. But there is an organism out there called Clostridium, right, which puts out a toxin that can cause your muscles to do this. And it goes into a complete tetanus. But this would be a graph of tetanus. You're up here, and there's absolutely no relaxation at all. The muscle is at maximum tension. So I've already kind of introduced this idea that when you are, those pictures I'm showing you there are more for individual muscle cells. Now let's talk about how you're going to use this idea to talk about your biceps brachii or any other muscle in your body. So remember that within any muscle, you typically have thousands of muscle fibers. And we've already described this idea of a motor unit. So going to your biceps brachii, you might have 500 or so motor units. 500 neurons sending signals down to a select group of cells within the biceps brachii. Motor units can be very small to very large. The smaller a motor unit, the more precise control you have over it. So your little eye muscles, those ones that twitch really, really fast, they have very few muscle fibers involved. You may only have 4 or 6 muscle fibers. So the motor unit is 1 neuron and 6 fibers, right? Very small number of cells. Whereas if you're talking about one of your big leg muscles, you may have a couple thousand muscle fibers. You would not have very precise control over your rectus femoris, but you would have a lot of gross control. So a lot of strength, but not a lot of precision movement. Now what we're going to see is that muscle fibers of different motor units are intermingled. So if I took a section of your muscle, I would notice that cells touching each other probably are not innervated by the same nerve or by the same neuron. These motor units are interspersed with one another. So here's a picture of that idea. So here we have, in this picture, 3 neurons. Okay? They're coming down as part of a nerve. And within that motor nerve, right, there's a bunch of, what's a nerve? A bundle of axons, right? And those axons, we got 3 different colored muscle fibers. We've got orange, blue, and purple. and you see that they're randomly associated within the muscle. So if I were going to lift something very light, I may only recruit the orange fibers to do the work, or I may only recruit the blue fibers, or I may only recruit the purple fibers, right? We're only going to recruit a limited number of the motor units to do a rather simple job. But if I needed to lift something very heavy, I would recruit all my motor units, and all of the muscle fibers would be contracting at the same time. So if you're going to lift up something light, I'm only going to recruit a limited number of motor units. The muscle will tense, but not every cell in my muscle is doing the work. Just a few of them are. Remember, muscle is elastic and extensible, so it kind of follows the lead of other cells. So you can stretch a muscle cell, right? It doesn't have to be activated to be stretched or to change shape. So here we see that the motor units are intermixed. So here's that idea of recruitment, right? First, when you're recruiting, you're going to recruit smaller units first. So I'm going to lift just a small pencil. I'm going to recruit, and the smaller motor units will be activated first, and then the larger, more powerful ones will follow behind. And what this is going to do is give a more smooth action to your muscle movement. Again, what do we know about smaller motor units? We have more fine control over them. So if I wanted to lift something moderately heavy, your body is first going to use the smaller motor units, a little bit more fine control over them, and then if you had to use more, it's going to pull in the larger motor units. Those larger motor units have more strength, not as much fine movement, but more gross motor movement in them, and that's going to contribute to a more smooth movement of your muscles. There is also an asynchronous summation. In other words, let's say I don't want to just lift up something, right? I don't want to just lift up this pen. May I borrow your pen? I don't want to just lift up this pen. Let's say it's a 100-pound pen, right? So I don't want to just lift up this pen. That would be recruiting for a certain number, right? I just want to pick up this pen and put it on the shelf. That's one kind of thing. But what if I just want to pick up this pen and hold it? And if this is something rather heavy, I would want my muscle fibers to take turns. So what happens is that within my muscle, there would be this asynchronous movement. So there would be a rotating basis of the motor units that are going to be recruited. Some would be working, then some would rest. And others would be working while the others are resting. So there would be this hopefully very smooth changing, right? Rotating of the motor unit so that I could sustain that contraction. Now we know at some point we get tired, right? But we're not talking about exhaustion at this point. We're just saying, oh, I've got a 100-pound dumbbell. I'm just going to hold it here for a few minutes, okay, or a few seconds. It won't be minutes. Okay, a few seconds here, right? And my motor units are going to rotate in order to do that. So imagine now that I want this much tension, right? The tension's pretty stable up here. But to keep this tension up here, I'm going to have an asynchronous but rotating assignment of the motor units such that it adds up to and maintains a relatively even tension. The practical part of all this is not difficult. We all lift stuff every day. We can appreciate this. What's new here is just the language, right? Using different words to describe these things. But we know that these are not uncommon experiences. So we can all appreciate what I'm saying. It's just we have not necessarily used this language to describe it.
So one motor unit does not work all the time by itself?
One motor unit does not work all the time by itself unless you are just picking up a feather, right? If I was picking up a feather, I may only, let's say, one or two motor units. That's all I need.
Sure. Okay.
But if it's something heavier, it's going to be more. And then if I need to sustain it, we're definitely going to be rotating. Okay? Definitely going to be rotating.
Now, how would you have more control over, let's say, like an eye twitch?
The smaller the motor unit, and by that means that I've got one neuron going to just 6 muscle fibers. It's a very fine movement. You've got a bunch of muscles in your eye. Remember you got 3 cranial nerves, right? 3, 4, and 6 that are controlling the movement of your eye. Those are very fine movements, Those are going to be relatively small motor units, some of the smallest motor units in your body, very fine control, versus your gastrocnemius or versus your rectus femoris, big 1,000 motor units at a time. Not very good fine control, but big clunky muscles, if you will. So you're asking control of that twitch. So when you twitch, it's very fast because there aren't many muscles involved. Go back to that graph where I showed you the eye twitch fast. Then there was, what was it? Gastroc and then something else, right? So the more fibers that are in the motor unit, the longer it will take to contract. The timing of that twitch will be different. So if I'm going to twitch my gastrocnemius or my rectus femoris, it's going to be a longer, slower process because so many fibers have to be relaxed and contracted, right? Contracted and relaxed in that cycle. Whereas the eye twitch,
That would make sense.
that make more sense now? When
I'm just thinking control.
when it twitches, you have no control, right? It drives us crazy, but it's quick.
The way, yeah.
It's quick because there's so few fibers firing, because that twitch is a single motor unit. It's one neuron zinging down to a limited number of muscle fibers. That's why the eye twitch is so fast. And where if you get a cramp in your leg, that's not fast, right? It's long, slow, laborious, and painful. Okay. Yes?
Do you make more motor units as you need them, or do you develop them?
They are developed. But if you're working out and you're creating more muscle, then you would also be creating bigger motor units. The number of neurons, when you work out, I'm going to increase the number of myofibrils, greater number of actin and myosin molecules, but I'm certainly not changing the number of axons going to my muscle. So I still have the same nerve going to my muscle. I still have the same number of neurons going to my muscle, right? But more muscle fibers would now be added to the motor units. So my bigger muscle has more force because my motor units are bigger. Each motor unit would be bigger in a developing muscle. But it's going to be a developmental thing that's going to happen.
So you don't lose them. You don't lose them either. Like, for example, if someone was overweight and then they lost a bunch of weight and they have all of these extra that have developed to pick up the extra weight, you don't lose them. You still have them.
Yes, yeah. But also if you had a muscle wasting disease or as you age and you lose muscle, you're going to lose those motor units randomly as well. So you're going to lose portions of this. Your overall strength would decrease, but you would still maintain your motor units. They would just be, each would be smaller and have less force that they could generate. Okay. That's in a normal pattern. Now, if there's some sort of disease state, it could be a little different, but in a normal, just aging phenomenon, as you age and you lose motor fibers, your motor units would become smaller. So when you tried to recruit and lift up that heavy thing, you wouldn't be able to as well as you did when you were 30, or if you were 60, okay? I've already mentioned the word tone before, so here it is again. Just there is a resting tension in your muscles. There is a certain number of your motor units that are always active, keeping your tension at that ideal length, keeping each sarcomere at its ideal length, that 2 micrometers, right, to keep your sarcomeres at their ideal weight, so that when you are asking your muscles to work, you're able to have the maximum force generated. So you've got to keep those little sarcomeres at that 2 micrometer perfect length in order to optimize your tension abilities. And this is all happening subconscious, right? I wish I could cause better tone, right? That's not something, this is something very much related to conditioning as well as autonomic. But like I said, it also eats up a lot of your energy. So good-looking people, it's not fair because they burn more energy just sitting there looking good. Okay. How are we doing on any of that conversation? Steven, you want to show off your muscles? Steven's back there. He's like, there's a little gun show back there. I don't know what's going on. Okay. Okay. Any other questions, though? Again, the daily... the story here isn't difficult, right? It's just making sense of it and asking good questions about what happens, but put your understanding of lifting a pencil to lifting a brick to lifting a car, and imagine what's having to happen in your muscles in order for that to happen. Question?
I probably know the answer to it, but my brother was a wrestler, so he's got all the muscle, he's ripped out. Well, he got a concussion, and he was out for about a month and a half, and he actually is bigger than when he was actually in the sport. Does that make sense? I mean, like, me and my family think that it's because his muscles had time to recover, but we don't really
Could
know.
be in part, right? And then that constant conditioning,
Yeah, because after he went
cutting
back
weight
to
and all that stuff. If
practice,
he's not worrying
he
about
got
cutting weight,
bigger.
yeah. Interesting.
Weird.
Yeah. Well, also though, when he was maybe when he was recovering, his diet changed a little bit too, and so you kind of shocked your body back into a different metabolic pathway.
He eats chocolate and pop and
I
everything,
know.
and he still weighs
I know.
135.
Those good-looking ones, they can eat all they want. I have to eat two. I have to eat two of everything, one for each hip. I mean, if I only eat one, I get unbalanced. Okay, isotonic. Okay, so when you have contractions, okay, there's some different motions here. Iso means what to you? Iso, same, equal, tonic. Right? When I say tonic, you're thinking tone, right? So isotonic is where there's equal or same tension. How could you show me isotonic? Isotonic would be lifting an object, anything where the tension has to stay the same. So I'm going to lift a 50-pound dumbbell, and while I'm lifting it, I have to keep the tension the same on all my muscles. So the same tension, isotonic. Now there's two different kinds of isotonic contractions. There's concentric and eccentric. Concentric and eccentric. Concentric is when you're rising against a load. So you're actually lifting the dumbbell, and you've picked up a dumbbell. You are actually lifting it. You're exceeding that 50-pound load. You're having to create more than 50 pounds of tension, aren't you? So if I have a 50-pound dumbbell and I'm lifting it up, that means I'm having to create at least 50 pounds of tension, aren't I, to overcome that. That is going to be an isotonic concentric. The muscle is shortening. The tension is staying constant. I'm just simply gliding and lifting up that 50-pound dumbbell. The speed of contraction is inversely related to load. So the heavier it is, the slower the contraction. Okay, so all I'm going to do is contract the muscle, and I'm lifting up the 2 kilogram pound. Okay, I was saying 50 pounds before, now it's just 2 kilos. Look at this graph. Okay, so what we've got here on the very top is muscle tension, and on the bottom is the length of the muscle, the percent of resting length. So the contraction begins here. Peak tension, tension is going to increase and then stay the same. This means isotonic, right? You see that straight line on the tension? Same tension, isotonic, straight across. What's going to happen? The muscles themselves, though, are going to do what? There's going to be fewer and fewer of them staying at resting length. And then the muscle will relax and everything will return back to itself. We'll compare that with the other types. And I said that they were inversely related as well. So here's the speed of the contraction versus the load. So if I'm only going to lift a 2 kilo, right, a relatively light thing, my speed can be a lot faster, can't it? But if I'm going to lift something that's 10 kilos, my speed will be slower. We know that, right? We just never saw a graph of this. So the heavier something is, it's going to take longer for us to get that contraction to occur. Now the other type of isotonic is eccentric. This is when the load is more than the tension that's being produced and the muscle's actually lengthening. Not only, if you're going to the gym, not only should you be trying to lift up dumbbells, but you should sometimes let very, very heavy weights pull your muscles, right? And you're trying to resist it, but you are not lifting it. So returning a dumbbell from a flex position. So you've got it, you're flexing your biceps, and now you're extending your arm. That weight is beating your arm, isn't it? Right? You have less tension on it, but you still are maintaining the same tension all the way down. And this is good. It actually elongates, and when this contraction ends, the lobe will stretch the muscle until there are tears in the muscle, and the tendons break, and the elastic recoil will oppose it. And there's some of that tearing that's necessary at the microscopic level for muscle to build up. So you have to kind of tear a little bit for the muscle to increase. So here, right, you're letting it go down. You're letting the muscle get stretched by that weight, but the tension is constant throughout. So again, if we looked at that graph, you would see that the tension goes up. There's that isotonic, right? It's isotonic. It's at the same tension, but you see that the muscles are actually getting longer, right? They're lengthening. Okay. Now that is isotonic. Two types, concentric and eccentric, versus isometric. What does that word mean to you? Same metric, right? Same distance, if you will. Think meters, right? Same meters, same distance. Now, this is where the muscle length is not changing. The tension never exceeds the load. This is your pushing against a wall. This is your pushing against your own hand, right? Everyone do the, I don't know what you call it, right? Take your palms and just push against each other, right? Push, push, push. Are your muscles working? Absolutely. Are they changing their length? No, they're isometric, but they're certainly doing some work, aren't they? And you're not going to push one hand over the other, right? They're the same, and it's like pushing against a wall really hard. Clearly, your muscles are working. There's some tension being forced there, and but you're not overcoming the weight. So this is just a postural muscle contraction. So as you're standing right now or sitting right now, your muscles are kind of doing that the whole time. They're in a isometric position. They're keeping you erect, your erector spinae muscles. They're not changing their length, but they are certainly doing a lot of work to keep it there. So here, right, we see isometric. Same weight. We're not overcoming the weight. The muscle's not lengthening. It's staying the same. Isometric. Graphically, right, we're going to increase the tension. Clearly there's tension, but we're not overcoming it. So you're pushing your hands. There's a peak tension, but the length of the muscles never changes. Right? Resting length stays the same. Isometric. Okay, so make sure you understand or appreciate isotonic versus isometric. And under isotonic, there were two types, concentric and eccentric. We know that energy is needed for muscles, right? ATP made by mitochondria. Your ATP is always being used up. You're always using up ATP. You don't store a whole lot of it in your body. There's very little free ATP. Your body's always making ATP. How is it always making it? What organelle? Mitochondria. And remember the old equation of cellular respiration? All right, so what's the equation of cellular respiration? The basic idea? Right, glucose plus, we better be breathing, right? Plus, sometimes you'll see water here. And what does that make? CO2 plus some water plus ATP, right? Some energy. So our cells are always always taking in sugar, and as long as there's oxygen around, we'll get lots and lots of ATP. And this is what all of our cells are doing. We say our cells are respiring, right? Cellular respiration. So we have a constant supply of ATP. The problem is there's very little free ATP. It's always being used up by your cells. Our body doesn't really store ATP in any one place. So the muscles, though, need to have a storage molecule for energy. And this is creatine phosphate. Okay? So creatine phosphate, you can buy it at GNC, you can buy it at the health food store. But this is the molecule that is going to supply enough energy for muscles to work for about 15 seconds. This is great for a sprinter, right? Somebody who's got to come off the block and sprint. ATP is not going to give you, there's not enough energy from ATP sitting around to do much more than about 10 muscle twitches. So the muscles have creatine phosphate, CP. That's going to allow some extra energy. Now, after the muscles immediately use up creatinine or creatine phosphate, then they have to rely upon glycolysis. And glycolysis is the breakdown of sugar that's done by your cells and done by your muscle. And as long as there's oxygen around, your muscles will be in a state of aerobic metabolism. They'll keep taking in oxygen, they'll keep making ATP. But if they keep working and oxygen becomes limited, then they'll kick over to an anaerobic state. I'm not going to get into this too much because I haven't spent much time in this course or last semester talking about glycolysis and about energy. I just want you to see the big idea that ATP is being produced from the presence of glucose and oxygen in your cells. Glycolysis does occur in the sarcoplasm, and glycolysis can be in the absence of oxygen. It does not require oxygen, so you can do glycolysis in an anaerobic state. That doesn't mean that you as an organism, that you don't have oxygen, but there isn't enough oxygen going to your individual muscles. You're working so hard at that moment that there's a lack of oxygen at the tissue level, and your cells can still make ATP, but only 2. And that's a pretty small number. And you'll also make some lactic acid and have that lactic acid burn. Most of the time, your resting muscles are going to get their energy from aerobic metabolism, and you're going to get a lot more ATP for this because you have plenty of oxygen around. So just a quick, this is just a big overview. Glucose comes into the cell. Imagine this big kind of orange box is a cell. It comes into any cell, in this case a muscle cell, into the sarcoplasm, and that glucose will make some ATPs and will, if there's oxygen, go down to the mitochondria and make even more ATP. So again, when there's oxygen, that's aerobic metabolism. It's going to use the mitochondria, and you're going to get more ATP than when there isn't any oxygen around. The other molecule, going back, the other molecule that your muscles are going to depend upon is glycogen. What is glycogen? It sounds way too close to glucagon, doesn't it? Glucagon was that hormone that was antagonistic with insulin. But what is glycogen? Can anybody describe to me what glycogen is? It's a long chain of glucose molecules is what it is. And our body does store glucose. It doesn't store ATP, but our body can store glycogen. It can store it in the muscles. It also can store it in the liver. So your muscles have some of this glycogen around, and then what can they do? The muscles can release the glycogen. The glycogen can then take the glucose and make ATP. So you've got some energy molecules sitting in your muscles waiting for you to recruit your muscles and for the energy to be utilized. So we've got some ATP. Again, you got ATP, you got the CP, and you got some glycogen. And this can keep the muscles going for a while. The more, you can train your muscles, right? The more exercise, you can train your muscles to be more efficient, and your muscles will actually store more of these energy molecules if you train more. I'm trying to think what I really want to say to you. We need the creatine phosphate. We need the ATP. Again, most of the energy stored in your muscles is going to be glycogen, and this is going to allow you an extended period of muscle contraction. I don't want to get into the nitty-gritty of this. At rest, okay, your muscles are not doing much. Their demand for ATP is low. There's still some demand, though. Because what are your muscle cells doing even at rest? They're maintaining some sort of tone, right? So even at rest, you're utilizing some ATP from the muscle sitting there. And at that point, you're getting energy from your bloodstream, and your mitochondria is making some ATP, and that ATP is being used to build up that CP and that glycogen. So if there is any extra sugar around or any extra ATP around, it'll be shuttled over to make that energy molecule creatine phosphate or make some glycogen. Now, here we see a blood vessel, and every one of your cells is next to a blood vessel, and there are fats and oxygen and glucose that come from your diet that are used by the muscle cell really to make ATP. Then that ATP can be used to make CP and also used to make glycogen. Don't worry about the specifics, but definitely we're making glycogen and creatine phosphate from the metabolism. Now, when you start doing some exercise, your ATP levels, your demands for ATP will increase. ATP production by mitochondria will go up, but remember that you only get a lot of ATP if there's oxygen around. So as long as there's enough oxygen, aerobic metabolism, you can go for a while. But at some point, if the demands are high enough, the oxygen will become limited to that muscle, and you'll no longer have it. And you will use up your glycogen, you'll use up your lipids and amino acids, and basically you will use up this immediate source of energy. As you go to higher levels, so we see here fatty acids and oxygen, and we're pretty much using up all the ATP here. There's not anything extra to make CP. Okay, so that's gone. And as we continue to work our muscles, there's going to be a huge increase in ATP demands, and your body can't keep up with that. So now you're going to go to the anaerobic, and you're going to start making ATP without oxygen through glycolysis. And again, this is not as, doesn't provide as many ATPs. The other thing it does, though, is it makes lactic acid. And lactic acid is an acid. And what do we know about acid? pH less than 7, right? And what that starts to do is it starts to decrease the pH in the muscle. And as it lowers the pH, it starts to affect enzymes in the muscle. And eventually the muscle cell and the entire muscle become fatigued. Muscles can't keep going. All right? They just can't keep going because of this lactic acid. Now again, the more you exercise, the more you condition your muscles, the longer you can go before you experience fatigue. So the more fit you are, the longer you can go. But at some point, you will reach fatigue. So at this point, you're using up all the glycogen in the cell, and you're basically making lactate. And that lactate is lowering the pH, a lot of hydrogens around, and that lactate is going to go out into your blood. Okay, so lactate's one of the waste products, and we'll talk about how our body gets rid of those acidic waste products when we get to the urinary system. So just want you to know the different energy molecules, ATP, creatine phosphate, glycogen, and that when your muscles are really, really busy working, that you're building up lactic acid, and that's lowering the pH. So at some point, you can no longer maintain the activity. The pH declines, and not only does the lactic acid goes up, but that lower pH starts to affect calcium troponin binding. You can start imagining what happens now, right? If you don't have troponin and calcium binding to each other, then you won't have cross-bridging going on in the muscle. And I've already mentioned that low pH also affects enzyme activity. So both of those contribute to fatigue. Also, we have lower pH from the lactic acid, as I mentioned, and the body temperature starts going up, and that's going to start triggering some sweating. This is not a surprise. And as that body temperature goes up, it can also start affecting some enzymes. So again, glycogen is released, and we're making lactate or lactic acid because there isn't sufficient oxygen. So what you see is that the mitochondria in this picture, it's hard to appreciate here, but the mitochondria is kind of faded out. The mitochondria is not being used here. You have to have oxygen for the mitochondria to do its job. So here the mitochondria is not being used, and the lactate's being formed. Then after you have done this excessive exercise, you've used up the energy, there's going to be a recovery period Now, this recovery period, I mean, if you're running and you get to a point where you're really exhausted, your muscles are burning, and you stop, obviously your respiration rate is up, you're going to be breathing heavily for a while. That's also part of the recovery. I'm just kind of talking about the muscle here. But once you start recovering, your body is going to huff and puff, isn't it? And that's going to help restore the oxygen levels. So the reason you continue to huff and puff after you exercise is to help restore the oxygen levels so they're no longer anaerobic, they return to an aerobic state. The more fit you are, the less you huff and puff, right? There's less of a imbalance. So then we'll go back to creating ATP through aerobic metabolism and get back to a normal place, and also the temperature of the body will decrease. Again, we know this stuff from just everyday experience. We're just throwing a few extra words into it. The lactic acid that was building up is converted back, and the pH also becomes less of an issue in the muscle cells during recovery. So you're just seeing now that as we're recovering, the mitochondria kind of takes back over, making the ATP, as oxygen now becomes available to the cell. So we're back to normal. We're back to rest. I'm going to skip over Cori cycle. So let's not worry about this Cori cycle or lactate cycling. So cross out this slide. Let me put an actual cross through it. Cross off Cori. Cross off the next one. Let's see how many we're going to cross off here. Cross off this one. We're just going to keep crossing off here. I think we're going to be crossing off till we finish up through this little section. This is all Cori cycle, so let's not worry about Cori. Okay. Last few minutes. Let's talk about the fact that in your skeletal muscles, you have different fiber types. So far, I haven't described that muscle cell. I mean, you know there's cardiac and smooth and skeletal muscle, but within skeletal muscle, there are three major types of skeletal muscle fibers. There's fast fibers, slow fibers, and intermediate fibers. At least the names aren't hard to remember. Fast fibers. The deal here is that these reach their peak tension very, very quickly. They're very large in diameter. They have lots and lots of myofibrils, lots of stored glycogen, very few mitochondria, but they fatigue very quickly. Okay, so the fast fibers store some energy. They can work very quickly, but they poop out fast. Okay, so that would be like your fast fibers are some of, for example, some of your eye muscles. They can fatigue. If you just keep looking your eyes up and down, that gets fatiguing, if you will. So those are fast fibers. Versus slow fibers. They are half the diameter. They're smaller. They take longer to contract. They can maintain a longer sustained contraction. And they're very, very red. They're dark appearing because they're full of blood and myoglobin. Now, myoglobin sounds like hemoglobin, doesn't it? Okay. Myoglobin is a special protein that carries oxygen to muscle. So hemoglobin, remember, is the molecule that carries oxygen throughout your body on your red blood cells. Myoglobin is a molecule similar to it, carries oxygen to your muscle. So this is a very dark staining muscle. It's darker because of all the blood storage. They are slower. They're not going to fatigue. So this would be your leg muscles, right? It's a good thing your gastrocnemius doesn't fatigue, right? You're walking all the time. It's very slow to fatigue. You're going to have a higher percentage of slow fibers in your gastrocnemius than in some other muscles of your body. Then the intermediates are kind of, well, as it suggests, intermediate. They're relatively pale. They're kind of in middle. They more closely resemble the fast fibers. Low myoglobin, kind of pale. in the middle as far as fatigue. That's why they're called intermediate. So that's easy. If you look under the microscope, you would actually be able to tell here these fibers are more white in color and these are more red in color. So which ones have more blood? They were the slow, right? And the more white ones would be more fast, right? So there's actually a color difference. You could see it under the microscope. We won't be seeing it this semester, but you would actually be able to tell the difference. Now, it turns out, if you look at this nice little table, look at this. This will compare for you fast fibers versus slow fibers and tell you about some of the characteristics that I just mentioned. In your body, you have a mixture of these fiber types. Your gastrocnemius is going to have more slow to fatigue muscle types or fiber types, muscles that are quick to respond but fatigue more quickly. There's going to be some differences. But there's also some genetics involved here. And there's some conditioning involved. So there's a little bit of evidence that if you are you can change the number of intermediate to fast with some athletic training. But much of it has to do with the muscle has what job normally to do and has also a lot to do with your genetics. So people who are fast, right? And part of it's their genetics. Part of it is just they can train to get a little bit faster. But a person who has a higher number of the fast fibers is going to be a better sprinter, right? Out of the gates fast, but they don't have a lot of endurance. And then people with more of the slow fibers would be greater endurance runners, long-distance swimmers. You can think about the differences. Some people just are better at distances. Some people are better at short sprints. And it has a lot to do with, again, genetics, what athleticism you inherited from your parents. And if you did a study of the muscle types within athletes, you would see that long-going distance runners would have a different combination of fiber types than a short sprinter. So it's really rare to find the person who can do both, right? When you find a person who can go short distances and long distances, that's a really rare person. Usually we're kind of specializing on one side or the other. So that's about muscle fibers a little bit. And what can happen to our muscles? Certainly we can increase the muscle size. So if someone's working out, what's happening? More myofilaments, actin and myosin. More myofibrils. That's actually going to make the muscle cell bigger. So the hypertrophy that you see is an increase in size, right? The muscles get bigger. Why? Because literally the cells got bigger. They have more mitochondria. They've got more glycogen. They're more able to do glycolytic. In other words, they can do more anaerobic things. They can go longer without getting exhausted. A person who is more fit, has more muscle, can work that muscle longer than a person who's not as fit. It becomes more glycolytic, more anaerobic. Again, the more you push your muscles to the edge, right, that's actually going to help this. Now, promoted by steroid hormones. We know steroid hormones do increase muscle. Guys, on average, have more muscle than women, in part because of their testosterone levels. Testosterone does increase muscle mass. And probably one of the only, I think, personally, reasons why a man should consider a testosterone supplement would be if their muscles are really beginning to atrophy and they're not able to maintain their muscle mass, that may be a reason to consider testosterone enhancement. However, right, we can't turn on the TV without hearing about blood clots and stroke and heart attacks from testosterone. So we know that for some people, the risk is worth it, but for the average person, taking more testosterone, right now we're looking at more health risks than good things. Atrophy is simply when the muscles are not being used. They're going to decrease in size and tone and power. Now, this can be just from a lack of exercise or a lack of use, but it also could happen, maybe there's a spinal cord injury, and so there's paralysis. And the muscles, if they do not have nerves going to them, will atrophy. Okay. Now, what does a person in a wheelchair do? They go to a lot of physical therapy. They try to have a lot of manual movement of the muscles to slow down the atrophy, right? Not that they're going to be using those muscles, but it will maintain their metabolism. If they can maintain more muscle mass on their body, then, well, they won't be able to, quote, use their legs, right, if they have a paralysis of the legs. But still, by moving those muscles, they can decrease the atrophy of the muscles, maintain their overall basal metabolic rate. You could, of course, have damage to the nervous system or spinal cord, and then you could have a fracture. So if you've been in a cast for a while, you know that your muscles will atrophy. Okay. If you're in a cast for 6 or 8 weeks, you take the cast off, you get back to your normal routines, your muscle will return. But if you're in some sort of spinal cord or brain injury, and there's a prolonged disuse, the muscle fibers will begin to die off, and they are not replaced. There are some diseases. Muscular dystrophy, Jerry's kids, we hear about these diseases. There's a whole bunch of them. They're all inherited diseases. They're all some genetic component to muscular dystrophies. There's something that disallows the muscle from working, right? So what does dys mean to you? Dys? Bad or painful, right? Doesn't work right. Dysfunction. And some sort of trophy, right? What is dystrophy? It's just not functioning right at all. So this is usually a muscle weakness, and in some cases we know that this muscle weakness can deteriorate rather rapidly. It's not only affecting skeletal muscles, but it starts to affect cardiac muscle, respiration, and some of these kids die rather young from pretty hideous circumstances. The most common type is Duchenne's muscular dystrophy, DMD. That is the most severe, or at least one of the more severe types. There's also Becker muscular dystrophy. It is a less severe form. So kids with Becker or people with Becker MD will live a lot longer. The progression is slower. The damage is not as extensive. But again, they typically die of respiratory paralysis just from the muscles no longer working. Polio. We don't hear about polio much anymore. But polio virus attacked the motor neurons, causing paralysis. So if your grandparents are living, ask them about, your grandparents should know somebody maybe with polio, okay? Maybe someone who, or maybe they heard stories from their own parents about people with polio. But this was pretty scary. I mean, a virus that spreads that can cause paralysis, and these people were put in iron lungs when they got polio. And if they survived that iron lung, they typically had some sort of paralysis as a result. And there were entire hospitals set up with big incubators and iron lungs to help them get through the polio infection. Pretty scary stuff at the time. Tetanus, I mentioned that tetanus of a muscle is when it's in full contraction. And Clostridium, a bacterium that lives in the soil, Clostridium tetani, puts out a toxin that can suppress your nervous system. And this is typically, you're going to get, this bacterium can live on something in the soil, like a rusty nail or something. And if it gets into your bloodstream, it can be very bad. That's why we get tetanus vaccines, so that we can play in the dirt and hopefully not suffer this disease. Has a pretty high mortality rate, so something you don't want to play with. So if you haven't had your tetanus shot in the last 7 years or so, it's time to get another one. Botulism, also made by another Clostridium, another organism. This one's food poisoning. But what that can do is this can cause paralysis as well. But look what it does. The toxin from Clostridium botulinum that causes botulism blocks ACH release. What would happen if you blocked ACH release? You're sending the signals, right? Your brain's saying go, 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 but you're not releasing the acetylcholine from the neurons, so what would happen? The muscles would be paralyzed, right? They wouldn't work. So there are any way that you can interfere with that whole story that I told you from top to bottom, there's a way of interacting or interfering with muscle contraction. And then myasthenia gravis, loss of ACH receptors. Again, what if you were sending the signal from your brain, acetylcholine was being released, but on the muscle, there was no longer an acetylcholine receptor. Same thing, right? Paralysis. So there's a lot of different diseases where these things can happen in the rigor mortis. Again, after death, 2 to 7 hours after death, the body starts going into rigor, and that's because you've depleted your ATP. What do we know about ATP? ATP is necessary for myosins and actins to disconnect. So the muscle is now rigid because ATP is not there. We come out of rigor depending upon timing, and we come out of rigor not because suddenly ATP is available, but because the body begins to break down and those protein connections are broken. So that's where I will stop with this unit, obviously. I will record those 2 lectures on the heart. I will post them early in the week or over the weekend. I'll send you instructions. Next week is break. The week after that, there will not be lecture. There will be these PowerPoints for you to listen to. There'll actually be 3 of them, right? 2 on heart, 1 on blood. And on the 17th, we'll have a review session. And on the 19th, we'll have our exam number 3.